And welcome back to the Infection and Immunity session for HCA Asia 2021. I hope you're following along all of the fun on Twitter. And I'd like to welcome you all on behalf of my co-organiser, Associate Professor Ponpan Matangkasombut. I'm speaking to you from Melbourne, Australia, which is traditionally known as Naarm. And I acknowledge that this is the traditional land of the Wurundjeri peoples of the Kulin Nation who've cared for this land for millennia. I pay my respect to their elders, past and present and future, and extend that respect to the Aboriginal and Torres Strait Islander peoples who joined us at this meeting, and to the traditional owners of the lands from which you join me today. Unfortunately, Ponpan can't join us today. She's literally a living example of the importance of cells of the immune system at work. And I have to say that's pretty rotten because she's been instrumental in creating a terrific program for today's session. So I'd also like to thank my Melbourne colleague, Ash Haque, for stepping into the co-chair role and assisting. So I'd like to waste no more time into getting into a wonderful program and to introduce our keynote speaker for the session, Professor Kazuyo Moro from Riken and Osaka University in Japan. Really excited to hear Professor Moro speak. She was one of the founders of the field of innate lymphoid cells, ILC2s, and she identified that lineage in 2010. And since then, her team has been elucidating the role of these innate lymphoid cells in a variety of diseases. Professor Moro, thank you for joining us. I hand the microphone over to you.
Okay, thank you very much for introduction. I'm Kazuyo Moro from Osaka University. Could you please share my slide? So I'm an immunologist, and I'm not a major, the single cell RNA sequence analysis is not my major focus, but I'm quite hard user of this technology. And so I would like to, today, I would like to, oh, someone is sharing her slide.
Can you share your slides, Professor Moro?
Yeah.
Are you able to share your screen?
Yeah. Can you see my slide?
Yes, that's perfect.
Okay, thank you. So yeah, so today I'd like to talk about our recent story about fibrosis in the lung. So this is my LAMB member. So my LAMB member are separated in Osaka and Yokohama, 500 kilometers far each other. So I'm now flying every week in this corona situation, but everyone in our lab are working on ILC2, which we identified in 2010. So all ILC2 do not express antigen-specific receptor, but they can respond to a variety of environment factors. So for ILC2, these factors are important for the stimulation. And once ILC2 get stimulated, they produce IL-5, 13, IL-6, and other cytokines shown here. And that's why ILC2 are now known to related allergic disease, because these cytokines are very important cytokines to induce the allergic diseases. But I believe that the original function of ILC2 was protective responses to the parasite infection at the time when we had a parasite infection. So when the parasite come into our body, they're gonna destroy the epithelial layer, and epithelial cells gonna die by necrosis, and necrotic cell gonna produce IL-33 and stimulate ILC2. So for ILC2, this IL-33 is very critical. And once ILC2 gets stimulated, they produce IL-5 and they induce eosinophilia, and this eosinophil can attack the worm. And also ILC2 produce IL-13 and induce mucin production from goblet cell. So same mechanism is used in allergic diseases, but in this case, allergen have similar enzyme as this parasite and automatically destroy the epithelial cells and make this epithelial cells release IL-33 and stimulate ILC2. But in the case of viral infection, ILC2 have good role because necrotic cell produce IL-33 also in the viral infection. But in this case, ILC2 produce IL-13 and act in the epithelial regeneration, and also by this filaggrin, they can support the tissue repairing. So that means ILC2 have variety of role in our body. So before moving to today's fibrosis story, I would like to show one data. So whenever ILC2 gets stimulated by IL-33, they immediately produce IL-5 and induce eosinophils. So this is a good model to show how ILC2 are important for eosinophil induction. This is IL-33 induced asthma model lung data. As you can see here in the wild type and RAG2 knockout, which looking T cell and B cell, ILC2 can induce eosinophils, but in common gamma RAG2 knockout mice, which looking T cell and B cell and also ILCs, there's no ILC2 and eosinophilia never occur. But if we inject, oops, so go to my slide. Go to next slide. Let me do it again. I'm sorry. Okay, now coming. So can you see my slide now?
You're coming on the presenter view. You might need to switch views.
I'm sharing.
Yes, that's great. Thank you.
Yes, okay. Thank you, and I'm so sorry. So when we inject ILC2 in the lung, we can now see the eosinophilia, which means that ILC2 is very important cell to induce eosinophilia. So this is the single cell RNA sequence data in same model, but as you can see here, you cannot find eosinophil. So this is ILC2 gate. Oh, I need to something wrong. Okay, this is ILC2, but you cannot find eosinophils. Even ILC2 is producing IL-5, and IL-33 expression is clearly high in ILC2. So this is kind of big stress for us that we cannot detect eosinophil by single cell RNA sequence. So hopefully in discussion time, I would like to have some discussion about why we cannot detect eosinophil by single cell RNA sequence analysis. So let's move to today's topic. So this project were done by Natsuko, who is excellent PhD student. So pulmonary fibrosis is a kind of horrible disease in the lung. The main symptom is dyspnea caused by the accumulation of collagen in the lung. And especially IPF is the most severe pulmonary fibrosis. And patient with this disease, IPF, are gonna die in like 2 years after diagnosis. So the reason why we start this project was kind of accident, because during 2016 and 15, we were focusing on the suppression mechanism of ILC2, because in many kind of diseases, ILC2 is doing something bad. So we wanted to learn what kind of factor can suppress ILC2. And we found interferon gamma can strongly suppress ILC2, and also the other group reported that Treg can suppress ILC2. That's why we established interferon gamma receptor RAG2 double knockout mice, which locking suppression mechanism of ILC2. So first we expected that these mice are gonna have a severe asthma, but after a while we noticed that this double knockout mice are gonna develop fibrosis, but not asthma. So Natsuko measured the body weight, and double knockout mice have lower body weight, and also checked the collagen production in the lung, and found that this interferon gamma receptor RAG2 double knockout mice have high collagen production. This is a CT scan data of double knockout mice, and we found that until like 13 or 14 weeks old, we cannot see any fibrosis, but after 15 to 16 weeks old, 100% of mice gonna start developing fibrosis here, and it's gonna be very severe when they reach to 37 weeks old. And so we count the number of ILC2 and also ILC3, and found that the number of both ILC2 and 3 are gonna increase at the age of like 16, 17, and 80 when the collagen production start, which means that ILCs might doing something bad in this double knockout mice. So we breed this double knockout mice with common gamma chain knockout mice, which looking ILCs, and the data was so beautiful because 100% of mice stop the fibrosis, which means that ILC1 or 2 or 3 might be causing this fibrosis. So we decided to carry out single-cell RNA sequence analysis by using this interferon gamma RAG2 double knockout mice. So I think more than 90% of previous work on fibrosis have done by bleomycin-induced or silica-induced fibrosis, but this is acute and temporal model, and we cannot assess what's going on in the pre-disease phase or what's going to happen in the chronic phase. But the good point of our double knockout mice is this is spontaneous model, so we can assess the differences in the pre-disease phase or chronic phase. So we got the cell from 7, 12, 16, 19, and 23 weeks old, and did the 10x chromics analysis. This is the data, and we could detect many different kinds of cells, and did the RNA velocity analysis, and 4 clusters show big differences: fibroblast and endothelial cells, DC, and ILC2 looks having a big difference during this fibrosis. And what we surprised was epithelial cells, which were reported to causing the fibrosis by DNA damage didn't show any difference in our model. So this is kind of surprising data for us, but this data suggests us to focus on ILC2. And fibroblasts were separated in two clusters, fibroblast 1 and 2, and we found that this fibroblast 2, showing gray color, express many genes related to idiopathic pulmonary fibrosis, IPF. So ILC2 is known to stimulate it by many different environment factors, not only cytokine, lipid, but lipid and neuropeptide and also hormone. And among these factors, IL-7, IL-2, and TSLP, and IL-25 and 33 is reported to be important for the stimulation of ILC2. So we assess these 5 factors. Unfortunately, we couldn't detect IL-25 in our single-cell RNA sequence model, but IL-2 and IL-7 expression didn't change a lot from pre-disease onset and to chronic phase. And TSLP expression looks getting lower during this disease. And what we think interesting is IL-33 expression become high during this disease. So we confirmed IL-33 expression by qPCR. And so in 7 weeks old or 14 weeks old, we cannot find the collagen production, but in this age, we can. And so we separate the tissue in normal area and fibrosis area, and we could detect IL-33 expression in this fibrotic area, but not in normal area, which means that IL-33 production is occurring in the fibrosis site region. So again, we made a triple knockout mice with IL-33 deficient mice and confirmed that fibrosis do not develop without IL-33. So next, to find out the source of IL-33, we use IL-33 GFP plus mice and gated on IL-33 positive cell. So it is well known that epithelial cells are the big source of IL-33, but we this time found that fibroblasts also expressing IL-33. So we check the IL-33 expression in epithelial cells and fibroblasts in different phase. And interestingly, the expression level didn't change in epithelial cell in 3 different stage, but in the fibroblast, the expression start at the onset phase and become higher in the chronic phase, which means that IL-33 from fibroblast might involve in the exacerbation of this the disease. So this is a subcluster analysis of ILCs, and here you can find that cluster 6, the number of cluster 6 is increasing from onset to chronic phase. And so we focus on this cluster 6 because the frequency of cluster 6 significantly increase from pre-disease to onset or pre-disease to chronic phase. And this cluster 6 was expressing IL-13, which is major fibrosis factor, and also IL-2 receptor alpha, which is a activation marker of ILC2, and IL-33 receptor, which is the most important receptor for ILC2 activation. And we finally co-cultured ILC2 with fibroblasts to check that if ILC2 can directly induce the collagen production from fibroblasts. So in IL-2 plus 7 condition, ILC2 can be maintain their survival, but cannot be activated. And in this condition, fibroblasts did not induce the produce the collagen. But in IL-2 plus 7 plus 33 condition, which induce ILC2 activation by IL-33, we observed high collagen production from fibroblasts, which means that ILC2 can directly induce the collagen production from fibroblasts. So the ineffectiveness of steroid treatment for fibrosis is often problem in the clinical practice. So we therefore evaluate the effect of steroid in different fibrosis stages. So when we start steroid treatment from age 24, fibrosis didn't stop. But when we start this treatment from age 14, when ILC2 activation starts, but still collagen production from fibroblasts is not occurring, we could stop the fibrosis, which means that if we can find out the fibrosis patient in the early phase, we can treat these patients by common steroid treatment. So I would like to end my talk with this beautiful human data. So we finally get the human IPF patient sample. We collect ILC2 from peripheral blood from 10 healthy control volunteer and 20 IPF patient. And this is a difference between two different patients. And ILC2s were separated, beautiful, in like a PCA, in like healthy control is here and IPF patient is here, except this patient, it looks very beautiful. And we also confirm IL-5 and IL-13 and IL-33 receptor expression in both healthy control and IPF patient. And it looks that even in the human fibrosis patient, ILC2 is highly related to this disease. So this is conclusion. ILC2 have a potential to produce IL-4, 13, TGF-beta, and filaggrin, which is all related to fibrosis. And once ILC2 start producing these factors, collagen production from this fibroblast can increase. And when this collagen producing fibroblast start producing IL-33, this IL-33 gonna give a feedback to ILC2 and re-stimulate ILC2. So we believe that that's why fibrosis is a chronic disease. And the steroid treatment analysis show that if we start the treatment from pre-disease phase, we can cure this severe disease. So I would like to thank all the collaborators. This work have done by Natsuko and for single-cell RNA sequence analysis, Tommy and Tom and Aki always help us. So I would like to thank them. So that's all. And I would like to take any question. Thank you for your listening.
Thank you very much, Professor Moro, for a really interesting talk. There are a few questions coming through. I'd like to start just by asking, did you see evidence of the IL-C2 cells producing TGF-beta in your model?
Okay, yes. Yes, we can. We just confirm by qPCR, and they do producing TGF-beta, but the expression level was not high, so high. So we're not emphasizing, not we cannot strongly say that ILC2 induce the fibrosis only by TGF-beta. So I think other factor, including IL-4 and 13 and amphiglin, is involved in the this mechanism.
Thank you very much. So the second question is whether ILC2 is also involved in airway modelling in other diseases such as asthma.
Airway what? Sorry.
So the question is, do you think ILC2 is also involved in airway remodeling in
remodeling.
asthma?
In asthma, I'm not sure in asthma. Other group have reported that they are involved in the remodeling, but I think remodeling function is more important in the virus infection because we get the data that in ILC2 knockout mice, the survival rate after flu infection gonna decrease a lot. So ILC2 is thought to be bad cell in the lung, but in the viral infection, I think ILC2 is doing very good role, and that is tissue remodeling.
Do you think ILCs are implicated in all types of fibrosis or just playing a role in the lung?
That's a very good question. So in human disease, pulmonary fibrosis can be divided in 8 or 9 different diseases, and we actually don't know if ILC2 is involving in all of the fibrosis, but because we just get IPF data, and other fibrosis are quite rare disease, so it's quite difficult to get the sample.
So I was really interested in seeing the longitudinal data, the time course that you had, and I wondered if you saw evidence of your stromal cells starting to adopt some kind of inflammatory memory,
Wow.
and whether or not. So how? I guess the question is, how soon do you have to intervene the IL-33, ILC2 treatment before that memory kicks off?
I have no idea for that question. Very good point, but sorry, I don't have any question. Sorry, answer for that question.
No problem, no problem. Thank you very much. Professor Moro, there's one more question relating to your intriguing observation that eosinophils were absent from the lungs of your model.
Yeah.
And I guess the question is this because you're not capturing them? And do you have any idea about why you're not seeing them?
People in eosinophil field say that the RNA of eosinophil can be easily broken, and because eosinophil have many enzymes, I'm not sure why this occurs, because we can get eosinophil and assess by FACS analysis, which means that cells are alive, but somehow we cannot detect in single cell RNA sequence data. So I want to get any hint for this phenomenon.
And I think that is a really interesting question to perhaps hold back to the discussion that we will have at the end of this session. So please join me in thanking Professor Moro for a really interesting and wonderful opening to the session. Thank you very much.
Thank you very much.
And our next speaker is Dr. Ashraful Haque, who is speaking to us from the Peter Doherty Institute for Infection and Immunity and the University of Melbourne here in Australia. So Ash moved to Queensland, Australia in 2006 to study immune responses during parasitic disease, particularly malaria. And he's been making forays into single cell genomics since setting up his group in 2012. We were lucky enough to recruit Ash to the University of Melbourne right in the middle of the COVID pandemic. His ultimate goal is to better protect against malaria and other infectious diseases by understanding T-cell responsiveness. So Ash will be speaking on defining the micro-anatomical change and immune cell interactions in the spleen using spatial transcriptomics. Ash, I'd like to invite you to share your screen now. Thank you.
Thanks, Chris, and thanks for the invitation to speak. I'll share my screen now. Oh, I'm going to stop that, try that again. I hope that's okay for you.
Right, thanks.
Okay, thanks for the invite to speak. It's rare for me to be involved in a group of people doing science. Usually we are, particularly in this region, debating who's got the best cuisine. It could be Japanese ramen, it could be Korean bibimbap, it could be laksa, the list goes on. But what's really lovely is for us to be working together towards a common good of generating a human cell atlas. So that's a really it's a pleasant aspect of the work and the initiative. So my interests are in the spleen, and the spleen here's a schematic, and it's known as a blood filter. So blood comes in, and blood goes out. But I want to draw your attention to the green area, which contains immune cells called T cells, and another yellow area that contains B cells. And these T cells and B cells must interact with each other, often to make antibodies that protect us against pathogens. And this is what it looks like in a mouse spleen, and the T cells are in bright red and the B cells are in bright blue, and you can see that they're juxtaposed. And in malaria, being a bloodborne pathogen, what's interesting to me is that over the course of an infection, and here on this graph we have spleen weight over time, day 4 to day 14, you can see that the spleen can enlarge by 8-fold, really quite rapidly. And so it's a highly dynamic organ. And not only is it dynamic from a macro perspective, the T cells within it, and the CD4 T cells in particular, are also highly dynamic. And in a study from last year, we studied CD4 T cells using single-cell transcriptomics and examined and generated a map that allowed us to look at specific genes. I'm showing you 2 genes in particular here in the video that are associated with a type of T cell called a T follicular helper cell. And these cells are thought to interact with B cells, are known to interact with B cells. But from the transcriptomic data, we can't really tell whether what we are seeing in bright red here in the slide are actually follicular. So we need to look in the tissue. So in order to examine that, and to do so not in TCR transgenic T cells, which we use to make the map, but really highly diverse polyclonal T cells, we started a collaboration with Fei Chen's lab at the Broad, who developed SlideSeq and a more highly sensitive version called SlideSeq 2, version 2, in which you have a circular array of 10 micron beads. It's about 3 millimeters in diameter. There's about 40,000 beads, all poly-T coated, so that when tissue is placed on top of the tissue, a localized mRNA capture can occur. You can amplify the library and sequence using next generation sequencing methods. And so with Fei's help, we performed this on 2 sets of samples. Uninfected spleens in bright green, and day 7, end of the first week for pink. And you can see in the bottom graph that the numbers of genes that we were picking up per bead was in the region of hundreds to maybe just over into the thousands. And that's enough that you can do unsupervised clustering. And for those of you who know your spleens, you'll be able to see that there's some splenic microarchitecture there with red pulp and white pulp areas. But it's important to remember that each circle is not a cell, it's a bead. And that when you place the tissue over that array, you may get two cells contributing their messenger RNA to one bead. And if that happens across the whole tissue, well, of course, that's a fairly complex situation where you need to then perhaps split the signal at every given bead into its component parts. But if you look at the real data, these donuts are from 4 individual pucks. And if you add up the yellow and the blue segments, these are inferred to be doublets based on existing single cell reference data sets. So more than half of the data is actually doublet or merged together. So what we need to help us deconvolute that data is a ground truth. So what you're looking at here is B cells produced by chromium 3' sequencing, and the object here was to take this ground truth, if you like, single cell data set and map it onto the spatial puck and therefore sort of localize these transcriptomes. So in order to do that, we again used some packages developed by Faye's lab and others at MIT. The one that we favored has been called robust decomposition of cell type mixtures. And that allows you to essentially split each bead into to model the transcriptome as a linear combination of different cell types. And by doing that, maybe we can see we can make sense of the data. So on the left, we took a stromal cell reference data set from expert colleagues at the Doherty, Professor Muller and Dr. Alexandra. And we could see there was structure when we tried to map those T cell, B cell, red pulp stroma cells into the array. And if you try and map a completely different reference data set, the B cell and T cell data set on the right, you can start to see some very clear structure that maps, that shows you that there's probably a lot of detail in these slide-seq version 2 arrays that can be deduced with the right references. So having done that, we really wanted to focus on 4 populations that are in circles on the left, 2 different types of B cell at the top and 2 different types of T cell state at the bottom. In particular, we're interested in the yellow population, and does it co-localize with B cells in blue? If we zoom in on these pucks, you can see possibly that yellow cells seem to be more associated with blue, and perhaps there's some suggestion that some green cells might be more associated with more of the red cells. So in order to quantify that, we essentially binned groups of cells in squares that are approximately 150 microns by 150 microns. And within those bins, you look for co-localization of transcriptomes. And indeed, if you take the cells that look like T follicular helper cells, they co-localize with B cells. They do not co-localize with plasmablasts, which are in a different area. So that's, before I get up to that point, so that's consistent with the idea that those transcriptomes that we had published were indeed bona fide follicular T follicular helper cells. We also noticed that T helper 1 cells did have a modest co-localization with plasmablasts. And that's something that we didn't really expected, and is therefore an example of something novel that we hadn't really expected to see in the data. Now, on these new maps of the single cell data, I'm drawing your attention in the top to 2 receptors which are supposed to receive, which are needed to receive interferon gamma signaling from those T cells that may be juxtaposed. And so we don't really know what this means, but it may be that plasmablasts insulate themselves from interferon gamma by downregulating it during infection. And we're trying to test that in the wet lab at the moment. So the last point I want to make is one that really is it's a very much a work in progress. But of course, using poly-T primers allows us not only to look at host messenger RNA, but we can also look for malaria parasite signal. And when we do that, we can see that, well, there certainly is a signal there. And if you look at the types of genes, types of parasite genes that we can detect down in the bottom left, it's over 4,000. So more than roughly 80% of the types of genes within the Plasmodium shivaldi genome can be detected. But if you look at the far right red column, the numbers of parasite genes per spot is very low. I mean, 5 genes or less per spot. So what we can really do with this data, we have no idea. That's why I'm putting it out there, just to show you that we can indeed find parasite. They're associated not with T and B cell areas, but certainly in the red pulp areas, as what one might expect, where erythrocytes are filtered out by macrophages. But as to what we can do with this data, it's very early days. So to sort of summarize, what we hope this near single cell resolution approach allows us to do is look at the structure of the spleen, to see how it changes, and to see how T and B cells change over time, and in particular to look for novel interactions between T cells and B cells. And I think I'm going to leave it there, actually, and thank my team, and in particular thank Cameron, who's on the left on the main photo, and is delving into a rather large cake on his birthday recently. So to thank him for all his work on this project, and be happy to take questions. Thank you.
Thank you very much, Ash, for a really beautiful talk. First question that's come through is just to ask you to elaborate a little bit more on this, what I know is very new data. How are you using the single cell data that you have generated previously to map onto these spatial data sets? Can you use that to deconvolute some of the mixtures that you've alluded to?
Yeah, absolutely. So each bead is modeled as a mixture, and you can use RCTD to either look for any number of cell types, which is only dependent on the number of reference cell types you have in your often a chromium data set or whatever other single cell data set. Or you can, as I understand it, although we have not done this, is to use RCTD in doublet mode. So you can restrict it to say I'm only assuming that there may be 2 cells at that bead, and so that's what we've done. And when you look at those pretty sort of images of beads and with different colors, they're often a composite of multiple probabilities of being, 10% T cell, 80% B cell, 10%, don't know, 10% some other cell type. Does that answer the question?
Oh, thank you. The second question that's come through is just whether or not you can identify specific germinal cell B cells, the LZs, DZs, germinal cell B cells from your data set, given that they're also presumably compartmentalized spatially.
Yeah, we'll be looking to do that. So in our experimental infection model, germinal centered B cells become more prevalent after around day 14. And so we're in the middle of trying to extend our initial study to those sorts of time points. We have generated the droplet-based data and can confirm that we can see plasmablasts and germinal center-like B cells, which are themselves sort of somewhat of a enigma when we're looking at the data, because they're rapidly changing, dying, and so it's probably going to be an interesting adventure to look for LZ, DZ B cells in that, but we certainly would hope to look at them spatially as well.
Thank you very much for a really interesting talk. We'll move on to the next speaker now. And so we were scheduled again to hear from Associate Professor Ponpan Matangkasombut and her colleague, Assistant Professor Varodom Sungnak. Unfortunately, as we mentioned earlier, Ponpan is not well enough to speak, so Varodom will be carrying the load for this talk. Varodom, I'll hand the microphone over to you. Slides look great.
Thanks so much for your kind introductions, Christine. Can you hear me okay? Yeah, I don't remember if you remember this, Christine, but I met Christine for the first time exactly 2 years ago in Addis Ababa, where we have, I think, probably the last, in-person HCA meeting. I was so nervous, before I gave my first talk, and Christine was the one who said, everything's going to be fine. She calmed me down. So for that, I'm forever in debt. So I really look forward to, a chance that we can get together again, perhaps in the next HCA meeting. So Christine mentioned that, sorry, go on.
I just wanted to say I've been fangirling you ever since. Go ahead. All
right, yeah, thanks so much, Christine. So I was just going to say that, as Christine mentioned, that this talk is meant to be given by Ponpan and myself, but because she's a little bit under the weather today, so I'll be standing in for her as well. So I'd like to mention upfront that everything I'm telling you today is from a close collaboration between my group, who are systems biologists, and Ponpan, whose group is mainly immunologists. So Ponpan, as mentioned, she's an immunologist, and she has been working on dengue, another mosquito-borne disease, in addition to malaria, for a very long time. But first, let us tell you a little bit about why dengue is a global burden, and why we should pay a little bit more attention to dengue infections. So in this map, you can see that the countries which are affected by dengue infections, and you can see that most of the countries affected are sort of developing countries that are situated around the equators. So this sort of tell me that this talk could fit into the inclusions and equity talk as well. Right. So, but in terms of like the number of people who are affected each year, there are quite a few millions of people affected, and even though most people don't show any symptoms, but still we have like millions, hundreds of millions of people with the symptoms, which means that they need to either be hospitalized or need to have the healthcare somehow. So really, that's quite a big burden. Unfortunately, right now, there's no antiviral drug for dengue at all. And there's only one approved vaccine with very limited efficacy. And also, it still has a lot of adverse effects in children. So clearly, we need to pay more attention into this. A little bit more about dengue infections. So perhaps similar to other viral infections, including COVID-19, most people affected don't show symptoms. However, for the symptomatic cases, for dengue, we have a mild version called dengue fever, or I'll just say DF. So in these cases, you have fever, headache, and viral-like symptoms and so on. For those who have a little bit more severe versions, we have something called dengue hemorrhagic fever, or DHF, which you start to have some plasma leakage and bleeding. And in very extreme cases, the patients could have shock as well. So we call this dengue shock syndrome, or DSS. So dengue has very interesting clinical cause. So the patients with dengue usually present with very high-grade fever for several days. So we call this febrile phase. And then suddenly the fever drops down, which is this what we call effervescence day, or we just call it DEF for short. And that's actually the start of the very critical period where you have a high hematocrit. sudden drop of platelets, and that's actually when you can have the shock. So patients could face dengue shock and die in that day. The problem is that right now it's still very difficult to predetermine when this death is going to be, because that's when the healthcare workers have to prepare to give the patients enough fluid and so on to prevent the shock. So quite a lot of research has been focusing on the febrile phase in order to try to determine whether there are any biomarkers that we can use as indicators that the death is going to happen, and we have to be prepared to treat the patient. So as I mentioned, that Ponpan, she's been interested in dengue infection for a long time. And several years ago, she was part of a consortium called Dengue, where they collect blood samples from dengue patients in Thailand, and I believe also in other countries in Southeast Asia as well. So they collect the blood, and also they store them as a frozen PBMC. So a few years ago, when we have the single cell facility for the first time, we thought it would be a good idea to try to analyze some of those samples. So in one of our first pilot studies, we took a sample from 2 patients, one with dengue fever, the other one with dengue hemorrhagic fever. So we have 2 patients, but we have a 4 time point for each one of them. So we have 2 days and 1 day after before the effervescence, or this is when the fever subsides, and also 2 weeks follow-up, which we use as a baseline. In addition to these 2 patients, we have the healthy controls, right? So that's our first pilot study. So we went on to do our first single cell profiling of these samples, and sure enough, it's PBMC. So you expect to see that the majority of cells are T cells, which is not surprising. But what was pretty intriguing for us is that if you expand it into different days, we can see some interesting trends, like we see T cells tend to be most expanded at the day that the fever subsides, whereas things like B cell or plasma cells tend to be at the peak at the day -1. So that's just 1 day just before the fever subsides. So of course we have 2 patients right now, but all the trends of how different cells populations going up and down incredibly consistent between these 2 patients. So of course, because we did the single cell RNA-seq, we can also look at different genes that expand and contract at different days in different subpopulation as well. So I'd like to take you through quickly. So we can see some obvious trends of certain genes with certain functions that tend to be temporarily upregulated. For example, we see the type 1 interferon signaling pathways and response to viral genes tend to be mostly expanded at the early days, the earliest day we have, which is 2 days before the effervescence, and tend to be most prominent in monocytes. And conversely, we see the transvaginal genes tend to be highly suppressed at the beginning, and then after that, it becomes more and more closer to healthy control. So that's the trend that we can see in all of the cell types. We also see cell type specific expansions. For example, in monocyte, we see cytokine productions, inflammatory response, and so on. But particularly, we see that at the early days. So without the time course data, we would not be able to do that. For B cells, we see, as you can imagine, B cell mediated immunities, but also we see some genes that are responsible for antibody productions, particularly at the day -1. So that's 1 day before the fever subsides. We also able to observe some of the transcriptionally distinct cell types as well, or at least clusters of cells. So one example I'd like to show you is the subpopulations of T cells that they express different genes profiles. The one that I'd like to highlight are the effector CD4 and subpopulation of effector CD8 that we call CD8 group 2. So these 2 subpopulations tend to have a lot of tissue homing markers, And the one that we particularly interested in is called CEL-PL genes. It's a skin marker, and it's encoded a protein called CLA. So in these two populations, interestingly we see these two expressions. We also see the expression of the gene called PD-1 as well, which is kind of interesting. So we went on to do a fact analysis with using a much larger data set. So instead of just 2 patients, here we have 20 patients, 2 time point, and also many more healthy controls. And we proved that that populations that have the expansions of the CLA proteins, we can actually see that particularly at day -1. So that's 1 day before the fever subside, and we see that in both DF and DHF cases, both in the CD8 and also in the CD4 population. So just to wrap this up, this is our first pilot studies that we have the first tackles to see the different genes, the different profiles that we can relate it to clinical cause of dengue. The most important one that we see is the skin homing signatures, which is kind of interesting because if you think about like dengue as a mosquito-borne disease, mosquito would bite, right? The patient at the skin, so it kind of makes sense that you have something that homes to the skin to respond to that infections, and we use facts to validate this particular cell cluster, clusters of cells that we think have the skin homing expansions in the PD-1 positive cells. Right, so that hopefully that would be the first of the series of what we're doing. Apart from that, our group are also working on other aspects of dengue infection, including using TCR and BCR repertoires. So the data I show you, we use 3'. So obviously, this day we can use 5' to do the clonality expansion studies. So we are already also doing immune profiles at different severities. So in the pilot study, we have the H and DFS, but also it would be interesting to see what's the difference between asymptomatic and symptomatic patients. So that work is being led by Waradon and Anunya at the time being. And also those T cell and B cell that we talk about, it would be interesting to see whether they actually dengue specific. So we're trying to do things like CytSeq to study this as well. So with that, I'd like to thank for your attention. So Ponpan is in the audience, so she would be able to help me answer any questions that I cannot answer, which is probably all of them. Okay, thanks, Christine. I'd like to
thank you so much, Varodom, for a really beautiful talk and really elegant to see the time course data providing really interesting insights into the phases of the infection. My question is whether you have any knowledge about whether patients have seen the virus before this acute infection and fever, and would that make a difference to the immune profiles that you are looking at?
That's a great question. So Ponpan would have been the better person to answer it, but let me try to have a go. So I didn't mention, but the 2 patients that we analyze now, we call it secondary infections. So actually, I don't know if it's the same for other viruses, but for dengue, usually primary infection would not give you such a big problem. But when you have the secondary infection of different serotypes, that's when you have big problems. So I cannot answer that question to you, but in our study we make sure that both of them have secondary infections. But I believe that that's another big problem that we have to try to find the differences. What's going on with the primary infections and what's going on with the secondary infections. But in our case, what we can do is just to show that they have another infection before. If that answers your question.
Oh, that's fantastic. There's another question coming through regarding the resident microbiome of the skin. Do you think the fact that there is a microbiome on the skin might play a role in homing of the T cells post-infection?
I can only say that's the great questions, but I don't think I know enough to comment on the resident microbiomes certainly we didn't look into that. So if it's okay, I will leave that to Ponpan to answer later on, if that's okay.
Absolutely. Varodom, thank you so much for a really interesting talk.
Thanks so much, Christine.
Thank you, Thank you, Ponpan, and the audience as well. I neglected to say in my introduction that Ponpan and Varodom established the first single-cell genomics lab in Thailand and have benefited from support from Sarah Teichmann's lab as well. I'll now hand over to my co-chair on the fly, Ash Haque, to introduce the next speakers in the session. Ash, over to you.
Thank you, Christine. It's my pleasure to introduce Assistant Professor Ishaan Gupta from the Indian Institute of Technology in Delhi. So Ishaan is a computer engineer by background, morphed into a molecular biologist, developing genomics tools such as single-cell isoform sequencing to characterize and understand the role of transcriptional heterogeneity in model organisms and human disease. And the talk today is Synchronized Plasmodium falciparum cell atlas, effects of heat stress in malaria and beyond. So Ishaan, we welcome you and pass the microphone to you.
Thank you for the kind introduction, and thanks the organizers for allowing me to speak about something which is non-human, which is Plasmodium, that does affect a lot of us. So I'm going to talk about the cell atlas in the Plasmodium falciparum, which is the parasite that causes malaria, one of the two major parasites, and how does heat stress specifically affect this parasite. A brief outline about the talk. First, I'm going to introduce you to the origin of heterogeneity for the Plasmodium in vivo. How does it behave, and how is the heterogeneity dissected in the context of the molecular biology of the parasite in vitro. Then I'm going to talk about our system, our experiment, whereby we are essentially simulating inflammation of the host, the febrile period. And we have three main findings there for the Plasmodium in the intra-erythrocytic developmental stage, or the IDC, that in a typical synchronized culture that is used for many in vitro studies, there is heterogeneity. Whenever there is a stress, there is redistribution of cellular states across the developmental time course, and the fact that temperature stress induces some interesting phenomena such as antigen switching and pushes the parasite towards more dramatic disease. So the parasite is an interesting, Plasmodium is an interesting organism which is adapted to live in two different hosts, the mosquito and the human. After it bites the human host, it has two life cycles. One, it's called the hepatic life cycle, probably something that Ash also showed. And then there is something called the intra-erythrocytic life cycle, where the Plasmodium is multiplying within the red blood cells. And this particular life cycle which is studied quite extensively, because this is where you would be having a typical blood smear to detect whether you have malaria or not. And right from the ring stage, which is where the parasite enters the blood, it goes on to different stages of differentiation and finally matures and becomes a gametocyte that can be taken up with the mosquito. So something which is very interesting about this life cycle is that it is somehow circadian. The circadian clock for this life cycle is tuned to the host. So every 24 hours, the cycle completes. It is modulated by specific genes, and it also correlates with cyclic bouts of fever. So we wanted to understand exactly how this thing happens. Can we actually dissect the genes playing a role in this process? So typically, when you are not trying to isolate Plasmodium from infected human blood, you take the human blood, sometimes our own, in the lab, and we synchronize using some chemical and physical means to obtain what's called rings, which are at the beginning of that intra-blood cell cycle. So this is where we get the parasites, and then we basically, in the synchronized version of the parasites, we essentially look at how the parasites is developing in the context of various molecular markers that it displays. So ever since the advent of single cell sequencing, this has been quite a race. A lot of efforts have been put into space, whereby there's the idea to create a malaria cell atlas, whereby you can say, okay, identify which genes are playing a role in this differentiation cycle, and can we actually identify some targets that can stop this cycle? And our efforts there are specifically focused towards the red blood cell stage. So what we wanted to answer is, if you have a synchronized culture, at least that's what we began with, and you treat it in heat stress. So when we obtain these rings after synchronization, we grow them for 6 hours at normal temperature and in heat stress. We put them through a single cell sequencing pipeline on 10x. Do we see some changes? So the first interesting thing that we found was that we do capture cells. So parasites can be captured, they can be sequenced quite well. On the 10x, we were able to capture about 12,000 cells. And contrary to what visual experimental confirmation would say, after 6 hours, we would expect just the trophocytes, which is basically this specific developmental stage, to be present. But to our surprise, about 70% of the cells, although they were trophocytes, but about 30%, they were developmentally distributed across the entire trajectory. So there were cells which were still stuck at ring stage. There were cells which were much more advanced in the trophocyte stage as well. And this was corroborated by cluster-specific gene expression. So just to be sure about what we are doing, we went back to the published malaria cell atlas data, which was in vivo data, and we co-clustered our data along with it. So on the right side, you can see in gray, you have the malaria cell atlas data, and we projected our data on top of this. And thankfully, most of the cell states that we are capturing here, when I say most, more than 95% of the cell states are present in the malaria cell atlas. And they are representing physiologically relevant states such as early trophozoites, rings, late trophozoites, chiasons, some jargon. But there are also some interesting things that are coming up, which is one particular state that is coming up, which is cluster 8, very small cells, which I'm going to talk more about in the remaining talk. So just to summarize the major findings that we had, the first finding was that the Plasmodium cells themselves redistributed across the entire developmental cycle. So if you could particularly focus here on the early and late trophozoites, so that's cluster 1 and cluster 2, there was redistribution in temperature stress. When we talk about the slightly smaller clusters, the clusters found here, cluster 5, 6, 7, there was high expression of stress response genes. So specifically in these clusters, not in the other clusters. So suggesting that these are the cells which are most sensitive to heat stress. And then again, there was this particular cluster, cluster 8, which was only found in heat stress. So when we look deeper into this cluster, we find something very, very interesting. So the cluster 8 has extremely high expression of clonally variant multi-copy gene families, such as VARs, TEVARs, and RIFINs. So just to give you an idea about what these things are doing. So whenever you have an infection in the red blood cell, the proteins of the parasite are processed, and some of these proteins, such as the VAR genes, which get processed to be exported to the cell membrane and act as epitopes. And what is currently the understanding is that these epitopes are essentially telling the immune system, activating it and interacting with it. So if there are too many of them, there could be some antigen switching happening and there could be activation. So this is something that marks these cluster 8 cells. But what are they actually doing in the developmental time course? To understand that, we did a pseudo-time analysis. And we found that cluster 8 actually forms an intermediate state between mass of the cells and a small cluster, which is cluster 7, that represents cells that are undergoing gametocytogenesis, which is required for the next cycle of infection for the next host to happen. And if we specifically look at both these clusters, we find the expression of certain master regulators of sexual development, such as AP2G. This gene is of high interest, people are currently trying to knock it down and trying to understand how it affects the Plasmodium life cycle is also a good drug target. Along with it, you have overexpression of certain transcription factors that are talking to this AP2G, and a lot of histone modifiers that could regulate this entire process in a temperature stress specific manner. So what we propose is that we have identified, say, a novel regulome, which is playing a role in stress-induced gametocytogenesis in Plasmodium. and these could be potential drug targets. So as a conclusion, I would like to end with a model, what we propose. So how we understand the Plasmodium life cycle currently can be captured by these, the black arrow figure, the circle. You have the rings, you have the trophozoites, early and late, and they can go back and keep on cycling to enhance the numbers, or they can just exit and cause gametocytogenesis and can facilitate the next infections. What we suggest, what we find, is that in the case of infection, not in the case of temperature stress, parasites, they achieve what's called a reactive state that somehow can evade the immune system, but also feed more into the gametocytogenesis pathway and can enhance the parasitemia. Now imagine this is happening over 24 hours. Every time you get a fever, this thing is pushed forward and more parasites are moving towards gametocytogenesis faster. There's also another state called the stress trophozoite state, which I haven't talked about, which are spurious states. So the take-home messages that we have from this short analysis is that when we expect in laboratory the cultures to be synchronized, they're actually not very synchronized in terms of developmental time, and they're extremely heterogeneous. When you have temperature stress, it induces certain hyper-reactive cell states, such as this cluster 8, that can manifest over the cyclic bouts of fever and can enhance malaria. And we suggest, and we have some data that is ongoing that suggests that actually this rewiring of development due to any form of stress could be a conserved mechanism in malaria, how it's trying to adjust because it has to adjust in 2 different environments. And the gene expression or genes which are playing a role in this rewiring could actually be revealing potential new drug targets. The stress trophozoite stage that I was talking about here is actually representing a very interesting state which expresses artemisinin-resistant genes, something that a lot of people try to study. And what we find is that this state is already present at very low abundance in both normal temperature and gets enhanced in stress. So this is something that has to be looked into when we are talking about bulk studies, which have previously been done, and a lot of stuff is already out there for that. So these could be contaminating these results. So I will stop here. I'll acknowledge my collaborator, Professor Krishnapal Karmodia, who has been doing all the wet lab work here, and his student Mukul, and my student Shreya. All the data that we have presented here can be interacted with and can be downloaded from the GitHub link. And we are hiring as well in both international postdocs and PhDs. Please visit the website, and happy to take questions.
Ishaan, thank you for a wonderful talk. I might get the ball rolling with a quick question. Could you tell us a bit more about how you projected your data onto the malaria cell atlas?
So we just did a co-clustering like the CCA in the Surat package. We tried a couple of things such as Harmony and there's another package, yeah. So we tried a couple of packages. CCA worked the best. It conserved the shape, and yeah, so that's what we did.
Okay, thank you for that. I was interested in also this suggestion that the heat stress induced this gametocytogenesis because you've captured the intermediate. Could you help us understand the expression of those highly variant genes, the STEVORs and the VAR genes? Does that, I should probably know the answer to this question, is that when you start to get shuffling of those genes and antigenic variation in malaria parasites? Is it at that point when this happens?
So, good question. We are trying to answer this by doing a time course. So this was just like 6-hour time point when the first trophozoite cycle happens. We are trying to do experiments now where we are propagating it for a much longer time, so we can actually pinpoint exactly when these events are happening. Currently, what we see is something which the field doesn't really believe in. So we are trying to convince people of that, is the fact that there is not a single VAR gene which is expressed per parasite, but multiple VAR genes that are expressed per parasites, especially when it's extremely stressed. And yeah, so yeah, to answer your question, it's an ongoing effort.
Okay.
We'll
I have to confess, I'm very super interested in your work, because we've just tried to, we've just posted a preprint of a mouse version of that, not looking at heat stress, but looking at inflammation stress. And we see some very interesting changes in the transcriptomes of individual parasites using the Chromium system as well. So maybe I can talk to you about that offline. But yeah, it was interesting to see that. So one last tiny question, which is, did you load infected red blood cells or all the red blood cells from your cultures onto the Chromium?
So we don't load red blood cells. We actually burst the cells with chemical treatment, and we load the parasites, actually, not the red blood cells.
Okay.
So we do have some contamination of human transcripts, but we can just just remove them.
All right. Thank you very much for your answers to our questions and for your presentation. We might move on to the next speaker in the session, who is Archita Mishra, and she joins us from Singapore Immunological Network at A*STAR, and is a Human Frontiers fellow. I just lost my notes there. And the focus is on early life human immunity, as we can see from the Da Vinci picture on our screens. And she's going to talk about a recent paper and it highlights human fetal immunity and microbial priming in early life. So Dr. Archita Mishra, I'd like to pass the microphone to you, and thank you for speaking.
Thank you so much, Ashraf, for such a nice introduction. And I would really like to thank the organizers, especially Christine, for inviting me. And I'm Archita. So today I'll be presenting our work on human fetal immunity and how the microbes might prime it. But before that, I would like to bring your notice to one fact that we as humans are basically constantly evolving ecosystem. We are partly human and partly microbial, which goes for bacteria, viruses, and all kinds of microbes. And they are very intricately intertwined with a lot of basic functioning of our body, which ranges from immune maturation, environmental tolerance, to nourishment and disease susceptibility. But the question that we asked in our lab is that when do we first encounter this functional microbiome, and how early is too early? When do we say that these microbes are actually friends, not the foes? So the decades of work in this field has actually shown us a kind of a picture what happens after birth, what happens during infancy, but there was a black box at the stage what happens during gestation, and whether there is any role of these microbes during gestation, and whether that can prime the development of our immune system. So with that question in our mind in Florence lab, and with our clinical collaborator, Professor Jerry Chan, we started curating human fetal organs, and these samples were very precious to us. And these samples were coming basically from the second trimester of gestation. And the organs that we were interested in were basically lung, liver, spleen, and gut, which is the gut, which is known to have a good and diverse microbiome, and certain organs which we know for the fact that don't have a particular microbiome of their own, like spleen or thymus. And we divided our study into three major portions. The first part was to basically understand how the immune system evolves during gestation. And the second part was whether there is any sort of microbe present in these fetal tissues. And the final part is basically the most important one, where we try to understand whether these two components ever interact with each other, the microbiome and the immune system. So first I'll talk about the first part, where we employed a very humble and very useful technique called CyTOF, which is again a single cell technique through which you can identify certain protein markers. And we employed a panel for T-cell specific panel of 40 antibodies. And we took the fetal organs and subjected to CyTOF. And to our surprise, what we found was that fetal organs, especially fetal lymph node, which is the mesenteric lymph node, and fetal gut, they had a very high amount of regulatory T cells. And these T cells were also high in PD-1 and Ki67 expression, which we concluded might be because of chronic TCR activation and active proliferation. And at the same time, when we looked closer to these cells, the pool of T cells present in human fetal organs, we saw a lot of memory T cells present in fetal organs. And these memory T cells were kind of effector memory in nature, like Ashraf showed in his work that the transition from the effector to memory, which is very important for any sort of to understand what kind of infection a body has seen and what kind of infection it can fight later. So here also what we saw was that these memory cells were effector memory in nature, and we try to hypothesize that what these memories could be against, because of, you know, a fetal environment is a kind of protective environment. Not much goes and comes out of it. So we wonder what could be these memory against and why they are so much inflammatory in nature. So to answer that, we hypothesized that there could be some microbes present in fetal tissues. And for that, we isolated these organs and subjected them to 16S ribosomal RNA sequencing, which I'll show you later. So the data that came out of these organs basically showed that there was a microbial diversity across fetal organs. If you'll see the exposed organs like skin, placenta, that is associated with the fetus, and the fetal gut and fetal lung, that was quite high in the microbial diversity, while the internal controls like thymus, spleen, and the PBS controls which we had, they were very low in microbial diversity. And we did this experiment at two places. One was in Singapore Immunology Network, where I work, and we also sent some independent samples as a blind study to our collaborators in Weizmann Institute. And at both the places, we got the similar result, that the diversity in the fetal organs increases and as compared to the PBS controls. And when we looked at the genera that we identified here, we could see that there were certain more than 11 genera which were specifically associated and enriched in fetal gut. But one might ask that what we are looking at is just the nucleic acid. So we also tried to culture these bacteria both anaerobically and aerobically, and we could find the similar genera present in the culture. And we concluded that what we are looking at is basically live bacteria. But as we all know, seeing is believing. So we wanted to really see where the bacteria is instead of like just randomly dispersed across the tissue. So we again collaborated with our excellent colleague Benoit, and we did scanning electron microscopy. And there we could see that there are certain regions in bacterial gut. So this picture that you see is an image of 14-week-old fetal gut. And we could see certain bacteria which were associated with the fetal gut inside the lumen. And there were certain regions where there was no bacteria. So we concluded that the localization of bacteria is basically associated with certain morphology within the gut, where you see more mucin-like structures. It is not just randomly dispersed, and it might have some role to play in the biology of fetal gut. And we did not see any bacterial presence in a 10-week-old fetal gut. So that was again another evidence that there is a time during which the microbes might enter the fetal system. Next, we also wanted to see how this localization pattern is arranged in the lumen. So we employed RNA scope technology along with our collaborators Ankur and Ria. And here you can see the green color basically represents the bacterial DNA, and the white is the epithelial boundary. And when you overlay that, the bacteria was quite nicely packed along the fetal lumen. And we could not find any bacterial presence in fetal liver or lymph node, which shows that basically what we are looking at is associated with the fetal gut only. And this brings me to the last and the most important part of the study, where we wanted to now see whether these two components, the bacteria and the more active immune system, whether they ever interact with each other or not. So for that, we devised the most syngenic system that can happen in such sort of study, where we isolate the dendritic cells and the T cells from the same fetal gut. And then we isolate the bacteria also from the fetus, and we present these bacteria to these DCs to present in turn to the T cells. And what we wanted to see was whether these bacteria can elicit a memory in these fetal T cells. And to our surprise, yes, we did find that there was a huge expansion in the memory component when we presented these bacteria, both in the fixed and non-fixed condition of the disease, which means that what we are looking at is basically both cytokine mediated and antigen mediated, but only antigen mediated itself had an impact on the emergence of memory cells which were happening in our system. And these memory cells that we found were basically active memory. They had CD69 expression, and I'm not showing you here, but they also had TNF and IFN gamma expression, which means they were effector memory cells, the ones which we saw earlier in the study. And we concluded that these are the memory cells which might be residing in fetal gut, which has seen the bacteria in the past. And when we recapitulate the system again, we can bring these memory back. And this paper was recently published, where we basically show that how the early life has, like the human fetal development does harbor some microbes, and how these microbes interact with the immune cells, and they basically have a memory in that compartment. Along with this work, we are also making a continuous landscape of immune cells, especially T cells, ranging from the fetal till the neonatal, adult, and even pediatric stages, using again CyTOF. And we are also using single cell technology along with the VDJ recombination technique to understand the tissue resident immune cells and how these tissue resident immune cells might help in establishing a tissue resident microbiome, because they, at the end of the day, they have to interact with each other. But the most important thing which I wanted to talk about today was this part, where we are trying to understand which immune cells interact with these commensal bacteria. This is still kind of a black box in the study, in the field, that how commensal microbes basically impact the immune system, and whether they are the key which help us differentiate between the good and the bad bacteria. And that's where the cell atlasing thing comes into picture, because when we are atlasing a human system, we are not just looking at single cells, because single cells are basically snapshot in time. They could be something else today and entirely different tomorrow. And what defines that is the environment which they are surrounded with. So with that in mind, what we are trying to do now is that previously that I showed you that we have seen the bacteria in the fetal lumen. So we employed another single cell technology, which is spatial transcriptomics, TSP, and we tried to understand where the immune cells are localized in the fetal gut. And to our surprise, the data showed us that there were certain regions, so these were the clusters, which showed us immune-rich environment. Some of them were rich in regulatory T cells, and some of them were rich in effector memory T cells. And there were certain regions which were kind of devoid of immune cells. So immune cells were kind of localized in certain spaces. And now what we are trying to do is we are trying to basically overlay these two systems and understand whether these bacteria, which we see in the lumen, and these immune cells, which we see through other techniques in the lumen, whether they ever interact with each other or not. And if they are interacting, what are those cells? And that will be another level of atlasing, where we are not just atlasing the human cell, we are basically atlasing the human cell in contact or in conversation with its environment, its microbial mates. So the overarching aim of our research is basically to understand the early life human microbiome, how that microbiome might affect the immune priming, and at the end of the day, the reason we all work is to understand how that can impact the diseases in early life or later in life, and how that can be intervened by better knowledge. With that, I would like to thank all our collaborators. The study was a massive effort across the globe, and a lot of fantastic people were involved in this. And I thank all of them, including Flohan's lab at SIGN, which was essential for the entire study. And I will be opening my own group in Telethon Kids Institute next year, and I'm looking for collaborations, and I'm very happy to talk to the fantastic HCA community and beyond to extend these work. And if any student is interested, I would really like to talk to them as well. Thank you. Thank you so much.
Very much for your talk, Archita. Couple of questions coming in. Christine Wells asks, do you think that the neonate's immune system could cope with the microbial assault of birth without having had this in utero training?
Yeah, that's a great question, because I mean, it's a kind of $1 million question, and that's why we want to understand this, that whether the fetal and neonatal immune system it's in itself ready to see the microbes that are coming outside. And that's where the conditions like preterm sepsis comes into picture, because if a baby is born preterm, we mostly see a lot of infections associated with these birth, while the full-term baby is not so much prone to infection. So I'll assume that is because that an important time during gestation where the microbes are coming and basically educating and priming the fetal immune system to make them ready to face whatever is coming outside. So I think fetal immune system in itself is learning, and as the neonatal immune system as well, because the environment changes. But these early components play a very important role in slowly priming the way towards more exposure or bigger exposure.
Thank you for that answer, Archita. I think we're going to move on in the interests of time. So perhaps you may want to answer some of the other questions in the chat if you can.
Sure.
But we're going to move on to our next speaker, who is Dr. Waradon Sungnak from Mahidol University in Thailand, who aims to understand the human immune system, especially in the context of infectious diseases. And the title of the talk today is Cellular Immune Responses to COVID-19 Deciphered by Single-Cell Multi-omics. So welcome, Waradon, and please do share your screen and take the microphone. Unmuting. Sorry, if you wouldn't mind unmuting for us, Waradon. I think your microphone is muted.
Oh, so I'm sharing the slide right now. So can you all see my slide? Oh, okay.
If you could put it on presenter view.
Yep.
Put slideshow rather.
So
that's perfect. Thank you.
So it's my pleasure to be invited to give a talk today. My name is Waradon, and I'm going to talk about my works during my time as a postdoc in Cell Attachments Lab in the UK. And the works revolve around cellular immune responses to COVID-19, deciphered by single-cell transcriptomics and multi-omics. We actually started our journey into COVID-19 studies way early, I think in February 2020. So at that time, the disease was still a regional epidemic. But we came up with a very, very simple question, which is that whether we could learn anything about COVID-19 even before we actually had a hands on COVID infected samples, given that we have a collection of human cell atlas datasets and as well as other single cell RNA sequencing datasets. So with that simple question, we just started to collect and compile single cell RNA sequencing data from different tissues. And then we check the expression of viral receptor ACE2 as well as expression of a protease shown to be important for viral entry, TMPRSS2. And even just by checking the expression and the co-expression, we found some interesting patterns, including the expression and co-expression of these factors in different surface barrier tissues, including in the airways, in the conjunctiva in the eyes, and in different parts of the gut, implying that this expression could be important for different modes of viral transmission. We also found expression of the viral receptor, even though, to some extent they don't co-express with TMPRSS2 in other tissues, including in the heart, in the liver, and in the kidney, implying that in certain contexts, viral infection could be important for the pathology in this tissue, and that requires further investigation, of course. But what we are mostly interested in is, of course, in is in the airways. And the reason that we got excited at the time was that during that time, it was widely believed that the receptor ACE2 itself was highly expressed in alveolar type 2 cell in the lung, conferring the disease pathogenicity. But what we actually observed was that once we look at the expression across the airway, we found expression of ACE2 as well as its co-expression with TMPRSS2, mostly in the nasal epithelia. And we think that this could have implications for viral transmission because of its high accessibility of the receptor in the nasal cells. And this is consistent across 2 different data sets. And we further look more into this direction by checking the expression of other viral receptors and entry genes in coronavirus that causes the common cold and influenza virus, as opposed to another coronavirus called MERS, which is limited in terms of its human-to-human transmission. And we found that for viruses that are expected to be easily transmitted, the expression of the receptor and the entry genes are more biased toward the upper airway, while the expression of the receptor for the virus that is more limited to human-human transmission is more toward the lower airway, suggesting that this could be a mechanism why some of the respiratory viruses are more efficient in transmission than others. And with this simple question, with some of these very relatively simple observations, it turned out to be a very impactful study with now, like just a year and a half after the publication, we got cited over, around 1,600 times, suggesting that, with just simple observation on what's already available through the human cell atlas, we could extract some of the useful information even before we actually had our hands in the actual COVID-19 samples. And that was our first step. And of course, after the disease became pandemic, then we actually got our hands on the actual infected samples. And that was when we started to be able to profile systemic immune cells at first in the blood. And this particular study was based on the collections of samples from 3 different cohorts from Newcastle, from Cambridge, and UCL Sanger, being composed of 130 donors with very detailed severity groups, including asymptomatic 1, mild 1, moderate 1, severe 1, and critical 1, based on the clinical phenotypes. And we collected the PBMC, and then we subjected the samples to 5' gene expression profiling, as well as TCR, BCR enrichment, as well as detecting the surface protein expression with an antibody panel composed of 190 antibodies. So we identify 18 cell types from the initial clustering, with additional 27 cell subsets after clustering. And I'm going to show you some of the key highlights from this study. So what struck us from the beginning was the enrichment of interferon signatures across almost all of the different cell types, particularly in the myeloid cells like monocytes and in the dendritic cells, as well as in hematopoietic stem and progenitor cells across different COVID-19 severity. We also found a very interesting monocyte that is CD16 positive, as well as expressing complement C1q. And we further characterize the cell by integrating this particular PBMC dataset with the bronchoalveolar lavage dataset that was previously available. And we found this particular monocyte to be in the transition stage based on the pseudotype analysis. So we hypothesized that this particular population could be a potential source of replenishment in the context of COVID infection. We also found interesting phenotype related to platelet. So first we found significant platelet myeloid interaction corresponding to elevated platelet activation signatures in COVID-19 donors. Also found increased megakaryocyte signature in the hematopoietic stem and progenitor cells. So both of them kind of pointed out to the direction that in COVID-19 donor, we both have differentiation of the blood cells into the megakaryocyte and platelet potential, as well as we see more platelet activation. And this could explain some of the phenotype, including thrombosis, which we see in COVID-19 patients. We also looked at the T cell subset, and we saw some subset that are associated with asymptomatic donors, including CD4 T cell expressing IL-22, CD4 T cell that are Th1 phenotype, and these could be associated with antiviral function, as well as providing the protection for the viral infection in the barrier surfaces. And what's also interesting is that once we subset the cell only in the clonally expanded groups, which is a soft, and I emphasize it's a soft proxy for antigen-specific activation, even though we could not rule out bystander activation. But even with this, we saw a trend of clonally expanded CD8 effector cells with disease severity, suggesting that having too much of effector CD8 T cell might actually result in a more severe immunopathology. While on the flip side, having more of the effector memory cells are associated with asymptomatic donors. And this could have implication on memory responses for the patients who have initial disease severity from the beginning. We also look at T cell or B cells, and we found certain interesting phenotype, including elevated IgA positive B cell associated with asymptomatic donors, especially in IgA2, and this could imply having secretory isotype providing protection in the actual barrier surface in the infected donor. We also found, based on the correlation of cell type frequency, we found a significantly positive correlation of circulating TFS cells and plasma cell only in asymptomatic and mild donors, suggesting that having TFH with plasma cell could lead to a better affinity maturation and then associated with better protection without causing, of course, immunopathology. So overall, we provided the resources, including the data set that we had in the first work and the second work, as well as a collection of other data sets that are ongoing at this COVID-19 cell@last.org. And we are open to having more data set being deposited on the website. And finally, I would like to acknowledge people being involved in the projects. For the first work, I'd like to acknowledge Sarah Teichmann, Kirsten Mayer, Ni Huang, who are instrumental in the initiation of the work, Martin, and Pascal Barbry, who are pulmonologists and immunologists specialized in the lung. And for the second work, there are a lot of people being involved in the project, including Emily Stevenson, Gary Reynolds, Rachel Botting from Anish Prasad, Fernando from Berto Gartens, Mike Morgan from John Mariani, and Kevin Tong from Menex Lab. So that concludes my presentation, and I'm open to any questions. Thank you.
Varodom, thank you very much for a fascinating talk. It was very impressive to see the speed with which your paper arrived and was published and was heavily cited by many people. So congratulations on that work. I wanted to ask you whether is it possible to look at a large number of patients now and correlate disease severity with any aspect of the epithelial cell response?
So there's actually, a follow-up work, even though, I'm not one of the lead authors, but I'm a co-author in that paper. So we actually look at the nasal cells, as well as the matched blood from both adults and children. And we saw some associated phenotype related to COVID-19. For example, in the nose, we found certain epithelial cell having inflammatory phenotype, so something that was mentioned yesterday as well, and also some differences between the local and the systemic one. For example, in the children, we found more interferon signature in the nasal tissue, while in the adults, we found more interferon response signature in the blood. And this might explain, why, maybe one of the reasons why children respond better than adults. So, this is an example of, some of the follow-up works that, not just us, but also authors were trying to do additional work in this direction.
Okay, thank you for that. A question coming in saying, do you think this increased platelet MAC interaction is important in COVID, important to the COVID pathology, or due to activated sticky phagocytes? I hope that makes sense. I don't think I did a good job there.
That I have no idea. Sorry.
Okay.
Sorry about that.
Okay, no problem. Well, I think we might move on if that's all right. And I'm going to pass back to my my co-chair, Christine Wells, to take over proceedings from here on. Thank you, Chris.
Thanks so much, Ash, for stepping up to the plate and helping co-chair. That was really much appreciated. So our next speaker is Dr. Melanie Neeland from the Murdoch Children's Research Institute in Australia. Melanie leads an immunology research program investigating early life immune mechanisms of allergic and respiratory diseases. She had a really very compelling paper published out today. Congratulations, Melanie, on pediatric COVID, but that's not what she's going to be speaking about. Here, she's going to be telling us a little bit more about mapping pulmonary and systemic inflammation in children with cystic fibrosis at single cell resolution. Over to you, Melanie.
Thank you very much for that introduction. Yeah, as mentioned, I'll be talking about cystic fibrosis today. So just a quick introduction to CF. It is a life-shortening autosomal recessive condition. It is commonly diagnosed at birth by newborn screening, and current estimated life expectancy is 44 years. It is caused by mutations in a gene which encodes for the CFTR protein, and this CFTR protein is expressed on the surface of epithelial cells throughout the body, and it regulates cellular chloride content and mucociliary clearance. And CFTR mutations result in the absence or defective function of the CFTR protein. And this then results in a multi-system disease. However, the major source of morbidity and mortality in CF is lung disease. So we know that CF lung disease evolves early in life, with structural lung disease in the form of bronchiectasis evident in the majority of patients by 5 years of age. We also know that airway inflammation plays a key role in driving this CF lung disease, and it's characterized by early non-resolving and harmful activation of the innate immune system that is seen even in the absence of infection. And it is this baseline pro-inflammatory state combined with recurrent infections that we see in children with CF that leads to progressive lung damage. And so for this reason, there is an ongoing search for targeted and effective anti-inflammatory therapies. However, other than what is performed on clinical testing, the immune responses in the lungs of young children with CF remain relatively unexplored. So to do this, we utilise a cohort established at MCRI and the Royal Children's Hospital in Melbourne, known as the ARREST CF cohort. And this consists of 400 children with CF. They are recruited at diagnosis, and following which they undergo annual bronchoscopies, lung CT scans, and bronchoalveolar lavage and blood sampling from 1 to 6 years inclusive. And so within this program, we collect paired BAL samples to sample the immune cells of the lower airway, as well as blood samples to investigate systemic immunity. And we apply a range of immunological assays to these samples that I'll be talking about today. So as mentioned previously, the immune response in the lungs of children with CF is not really investigated previously. So in order to do this, we had to establish a flow cytometry panel that allowed us to phenotype all major immune cell populations in the BAL. And so we did this using our 5-laser Cytek Aurora at MCRI. And what we showed is that the major immune cell present in BAL are alveolar macrophages, and they sit at about 70% of immune cells in the lung. And these can be phenotyped by a range of markers such as CD115, CD206, and HLA-DR. And we also show that granulocytes make up a large proportion of immune cells in the BAL in CF as well. And these are phenotyped based on pan expression of CD15. However, they can also be subsetted further based on expression of CD16 and CD66B. Additionally, we show other innate cells present in the lung, including dendritic cells and 2 subsets of monocytes, as well as cells of lymphoid origin, including natural killer cells, NKT cells, CD8 and CD4 T cells, and B cells as well. As we collect BAL and blood samples from the same participants, we're also interested to compare the immune cell profile in early life across respiratory and blood compartments. And so we've done this here on the left-hand side using unsupervised hierarchical clustering. And what this does is pull out the BAL cells here in blue quite nicely from the blood samples in orange. And that's based on the immune cell profile of cells listed here. In particular, this was characterized by higher proportions of cells of lymphoid origin in the blood, including B cells, CD4 and CD8 T cells, as well as NK and NK T cells, and higher proportions of innate cells, including CD16 expressing monocytes, granulocytes, and dendritic cells in the BAL compared to blood. And this was quite consistent in each individual, as shown underneath here in the line plots. So far all the data I've shown has been in children with CF, but one of the key questions is how does this differ from a healthy state? And that's actually quite hard to investigate in the lung, as healthy children don't necessarily undergo bronchoscopies, which are what is required for collection of the BAL samples. So with help from our great clinical team, we've been able to recruit 10 healthy control children who are undergoing bronchoscopy for reasons such as stridor or snoring. However, they have no history of lower lung disease. And so when we compare this cohort to our cystic fibrosis cohort, we see increased proportions of all subsets of our granulocytes in children with CF compared to healthy controls. And this is also reflected in reduced proportions of NK cells, CD8 and CD4 T cells in children with CF. We can also see this below on the UMAPs, which this here just represents an unsupervised flow-sum clustering analysis, where we see infiltration of 3 subsets of granulocytes in the children with CF. As we also collect samples throughout the first 6 years of life, we wanted to see if there was any differences in the immune cell profile between our early life samples, so our infants and preschoolers, compared to our older children, so from 1 to 6 years of age. And what we've actually been able to show is positive correlations between positive CD66B low granulocytes in the BAL in children with CF and age. And we also looked at this in the blood and showed that neutrophils increase with age in children with CF. However, CD4 T cells and B cells decrease with age. However, how does this compare to healthy controls? We also looked at this, and what we show is that children with CF have this marked increase in granulocytes over the first 6 years of life. That's not seen in healthy control children. And we also see this sustained reduction in CD4 and CD8 T cells in children with CF. So in addition to the immune cells, we're also interested in soluble mediators of inflammation. And so we measure this in the BAL cell-free supernatant by multiplex beta ray. And what we show is a range of inflammatory cytokines present in the BAL of children with CF. And these actually quite highly positively correlate with proportions of granulocytes. And so this then further highlights that positive feedback loop between increased inflammatory cells and then increased inflammatory cytokine and chemokine production as well. And if we then compare these inflammatory cytokines in CF to healthy control children, we see marked increases in a range of these inflammatory mediators, particularly those involved in the recruitment of cells to tissues. So with that in mind, we wanted to also investigate the immune cell profile in CF in greater detail. And so to do this, we used a site-seek approach where we collected BAL samples from 8 children with CF, and they were aged between 5 to 6 years. And so this was actually a pilot study to see if we could get this site-seek workflow up and running, as this had not been previously applied to samples collected from children. And so in this experiment, the samples were cryopreserved and then thawed on the same day, and then they underwent live single-cell sorting, and then they were hashtagged and pooled and stained with the Total-seq A human universal cocktail from BioLegend, which contains 154 immune-related proteins. And then they underwent 10x chromium capture and sequencing at the single-cell open research endeavour at WeHi. In 7 of the 8 samples, we were also able to perform matched flow cytometry analysis using the spectral panel that I described earlier. So to annotate our site-seq data, we've actually been using a reference data set, the human lung data set that's included within the Azimuth pipeline. And this revealed immune cell populations that we'd observed previously by flow cytometry analysis, including large proportions of macrophages, dendritic cells and monocytes, our lymphoid cells, so CD4 and CD8 T cells, natural killer and natural killer T cells, and our B cells as well. However, what this analysis revealed to us, which we hadn't observed previously using flow cytometry, are 3 subsets of airway epithelial cells, which are the ciliated goblet and basal cells, as well as plasmacytoid dendritic cells and basophil mast cells. This is the individual participants. So each UMAP represents an individual participant on the top here, and then underneath are the proportions of immune cells identified in each of the participants. And as we were able to perform that matched flow cytometry analysis of the same samples, we could compare the immune cell proportions obtained using the 2 technologies. And in general, what we see is a really nice positive correlation between the cell proportions identified in site-seq compared to those with our flow cytometry panel. Interestingly, we do lose some of our granulocytes that we can detect by flow cytometry. They're not detected in the site-seq, which has previously been described. As we performed site-seq analysis, we could also look at the protein expression in each of these clusters. This is just an example of about 60 proteins that we looked at. And what we see is nice expression and clustering of myeloid markers in our macrophage, monocyte, and dendritic cell populations. We see expression of T cell markers on our CD4 and CD8 T cells, as well as nice differentiation between our natural killer and natural killer T cells. We see markers of B cell and B cell activation for our plasma B cells. And the 3 subsets of airway epithelial cells cluster nicely, and they express markers associated with epithelial cells. As a further validation of our clustering or our cell annotation, we also looked at some key inflammatory cytokine genes within each of our cell clusters. And interestingly, we show expression of innate cell trafficking and granulocyte activation cytokines and chemokines in our myeloid cell clusters. We also show cytokines involved in T and B cell homing in our lymphoid cell clusters, and then cytokines known previously to be involved in airway epithelial cell function in our 3 subsets of airway epithelial cells. So in general, I think we're really happy with how the cell annotation has performed, and we're able to identify quite specific cell populations as well as their gene and protein expression. However, in addition to the main cell populations, we're also interested in subclustering our alveolar macrophages. And this is because alveolar macrophages make up 80% of the immune cells in BAL, and we know that they exist in different states, activation states in vivo. So this is just a preliminary analysis where we subclustered the alveolar macrophages, which revealed 22 cell clusters, which we've now been able to annotate based on marker gene expression as well as gene ontology analysis. And so what we've been able to show are macrophage subsets associated with cytotoxic activity, responses to bacteria, and the viral response as well. So just to summarize, we defined the immune cell profile of BAL from children with CF, and we performed a matched analysis of pulmonary and systemic immunity. Our work in progress is to expand this work beyond children with CF into other diseases such as asthma, wheeze, and healthy controls. And I might just actually stop there, so thank you.
Really beautiful study. Thank you so much, Melanie. And exciting to see that level of comparison between the flow panels and CytSeq and the RNA-seq as well. And so I noticed that you did comment that there was a high degree of annotation correlation between the various methods, but can I just push you a little bit further on that and ask whether there were any surprises? Were there any cell subsets that had poorer correlation than you expected?
With the gene and protein?
Yeah.
Yeah, that's a good question. We did show really nice correlation. And yeah, nothing that was poorer than what we expected. So we're still sort of doing more detailed analysis on that. But in general, yeah, the genes really did nicely correlate.
Can I also ask about the observation that you were seeing some of the granulocyte populations drop out of the 10x profiles? And I think you're right, others have noted that too. But do you think you're capturing all of the cells in the FACS panel as well? Because I noticed that there weren't any eosinophils, for example, in the lung samples that you talked through, and that had been mentioned by an earlier speaker.
Yeah, that's a great question. So for the FACS panel, we do run the samples fresh, so we expect there to be much higher proportions of granulocytes than there would be in the cryopreserved and thaw samples that we run on CytSeq. So that's one reason for the difference as well. In terms of eosinophils, well, I haven't looked at specific markers like Siglec-8 or something like that that would, specifically detect eosinophils. I think they're quite hard to find in the lung just because they don't really sit traditionally as they would if you compare it with a whole blood sample. But that's something we're looking into as well.
Thank you so much, Melanie, for a really interesting talk. I'm looking forward to the next chapter of that study, that's for sure.
Yeah, thank you.
So, it's a great pleasure now to welcome our final speaker for this session to the stage, Dr. Anunya Opasawatchai, who is also coming to us from the Madal University in Thailand. And Anunya is an immunologist and a dentist who is interested in the application of single-cell transcriptomics to dental infectious diseases. And that will be the topic of her talk today. Anunya, I'll ask you to share your screen. I'm just going to presenter mode. Am I going to presenter mode?
Yeah, 1 second, sorry. I'll share screen.
Sharing your screen. Just need you to put the presentation up.
Okay. Okay.
Great, perfect. Go ahead.
Okay, so thank you very much for your kind introduction. And first, I would like to thank the organizing committee for the opportunity to be here at HCA Asia. It has been fantastic so far. So today is my great pleasure to be sharing with you our work on single-cell profiling of dental pulp in health and disease. And it's one of the early works that we generated data in Thailand. So let me start with a brief introduction of the biology of the teeth. So the teeth are actually an interesting part of the human body because they are comprised of hard tissue, which are enamel and dentine, that encapsulated the soft and cellular part, which is called dental pulp. And being as the teeth have non-shedding surface, they are like the perfect place for the bacteria to colonize. And these bacteria are capable of producing acid, and those acid can demineralize the enamel and the dentine, and the bacteria will start penetrating deeper and approaching the pulp chamber. And once the bacteria get close to the pulp chamber, or when the pulp chamber is exposed, that's when the patients experience to sensitivity and pain. So the dental pulp, as it is it is kept in a confined space. So the inflammation in the dental pulp is not easily resolved. So in response to mild insult, such as slow progressing dental caries, and the odontoblasts, which are the cell types that produce the dentine, can generate what is called as reactionary dentine to wall off the infection. And the inflammation will be kept at minimum in this stage. However, in some other cases where insult is quite severe, that can result in the death of the odontoblast, and excessive inflammation often follow. And when you have excessive inflammation in the dental pulp, it can result in a toothache, a pain, and sometimes pulp, what we call pulp necrosis, which is the death of the dental pulp. So that leads to our questions. So we want to profile the dental pulp in a healthy stage and in deep dental caries, and to see how are the differences of the cell populations. And we are also interested in the regulations of the genes in response to dental caries. And we are also curious about how each cell in the dental pulp interact to each other in homeostasis and in response to deep dental caries. So here's our experimental outline. We collected permanent third molar, or better known as wisdom teeth, from the patients, and we collected 4 of them. So one of them is a sound tooth, and another one had enamel caries, and the other 2 had deep dental caries. So we performed tissue dissociation and enzymatic digestion before single cell library preparation by 10x, and we send them for sequencing. So these are our results. We were able to identify various cell populations in the dental pulp based on their canonical markers. Among them, we found odontoblast cells, and actually we found like a heterogeneous populations of odontoblast cells expressing different mineralization genes that are related to mineralization, such as OMD, ALPL, and DMP1. And apart from that, we were able to identify Schwann cells and glial cells, hematopoietic stem cells, endothelial cells, and also immune cells. So we want to compare the populations of the cells in the dental pulp between sound tooth, enamel caries, and deep dental caries. So with the UMAP plot here, you can see that the sound tooth and enamel caries share a pretty similar profile of the cell populations, but you can see these extra clusters of cells in deep dental caries. And these extra clusters of cells are actually B cells, plasma cells, and CD103 positive dendritic cells. So actually during homeostasis, some immune cells are already in the dental pulp, such as T cell, macrophages, but these B cells, plasma cells, and CD103 positive dendritic cells are only infiltrate the pulp in response to the deep dental caries. And so next we want to know how are the genes differentially expressed in the presence of deep dental caries. And here we, as the profile of the cells of the sound and enamel caries are similar, so we group them together and we compare the gene expressions against deep dental caries in each cell type. And as you can see here, so the blue circles represent the average expressions of the genes in deep dental caries, and the red circles are from sound and enamel caries. And these are the names of the genes that are differentially expressed between both of the conditions. And we can largely categorize them into 3 modules of the genes. So the first is the genes that are associated with inflammation. So we found NF-kappa B and IL-6 expression, which is upregulated in the odontoblast cells and endothelial cells and hematopoietic stem cells also. And we found genes associated with immune cell activations in the immune cells populations. And then we also found the genes that are related to mineralization that are upregulated in the odontoblast cell subsets, especially on the OMD positive odontoblast. And apart from that, we also found the genes that are associated with resolution of inflammation, such as ApoE, which were upregulated in the odontoblast. So in the later part of our studies, we want to predict the intercellular interactions of the dental pulp by comparing between sound and enamel caries versus deep dental caries by looking at the expression of the ligand receptors by the software or the package called NACME. So this is interaction network. As you can see here, each node represents each cell type, and the edge represent the possible interactions between the ligand and the receptors. And the blue nodes represent the higher expressions of ligands and receptors that are present in deep dental caries. And the yellow nodes are had very slight difference between the two. So you can see that in deep dental caries, the overall cell-to-cell interactions increase in many cell subsets, especially those of the immune cells. And the heat map tells the similar information, showing the ligand and receptors interaction. And because B cells and CD3 positive dendritic cells were only present in the deep dental caries, so you can find like the increased number of interactions in these cells with all the cell types that are present in the pulp. And you can also see the increased interactions between macrophages in like almost all of the cell types, and also fibroblasts with our immune cell subsets, as well as odontoblasts with macrophages. Okay, so then we look at each specific interaction. So we want to compare each ligand receptor pairs between sound and enamel caries and deep dental caries. So here are the top interactions between them. And actually we found that there are quite a few common interactions that were present in both of the conditions, such as the MIF and CD74 interactions. So the MIF was expressed by odontoblast cell subsets, and CD74 was expressed by CD16 positive monocytes. This is an example of the common interactions between the two. But we also found a specific conditions that were only enriched in the presence of deep dental caries. That is CCL2 and ACKR1. That is one, which is expressed on the macrophage and the endothelial cells. And also another interactions is between TIM1 and CD63, which are expressed by the odontoblast and the myeloid cell subsets such as macrophages and monocytes. So here we give an examples of the interactions that we talked about in the previous slides, showing that the CD74 is being expressed by the macrophages and B cells, while the MIF was expressed by the odontoblast, and also the TIM1 and CD63 that are being expressed by the TIM1 being expressed by the odontoblast, and CD63 being expressed by both the macrophages and odontoblast. So to summarize, we found the alteration of immune cell populations in the dental pulp, in the deep dental caries, and we found the upregulation of genes associated with inflammation resolution and mineralization in deep dental caries, and CCL2, ACKR1, and TIM1 CD63 are the interactions that were enriched in deep dental caries. And with that, I would like to thank the finding bodies and my mentors, Dr. Varodom, Dr. Ponpan, and Serintip and Benjamin Point, who play an essential role in computational analysis. And with that, I will be happy to take questions. Thank you.
Thank you so much, Anunya, for a really fascinating and deep dive into teeth. Can I ask, given that you're finding these new associations of dendritic cells and T cells deep in the dental caries, does that offer any insight into treatments or interventions?
Thank you very much. So that is a very important question, because normally because of the excessive inflammation that I mentioned, most of the time when the tooth reaches that stage, we often have no choice but to remove the dental pulp totally. So here we want to provide the information of the candidate kind of genes that we can possibly target to inhibit, to prevent excessive inflammation. So at least we can preserve the pulp in case of any inflammation. But that is to be further explored.
Can I ask whether you have much information about the type of pathogens that are feeding that interaction deep in the dental caries?
The type of pathogens? Okay, yeah, so that's a great question. So in dental caries, actually the pathogens are very dynamic, because at the early stages, there will be our oral microbiota, just oral streptococcus, which seems to be harmless. But when the cavity starts to get deeper and deeper, so the oxygen will become more limited, and that's when anaerobic pathogens will start to penetrate inside the dental pulp cavity. So just to summarize, most of them are oral microbiota, and when the condition favors their growth, there is a kind of dysbiosis that favor the growth of the bad ones.
Thank you very much, Anunya, for a really fascinating talk.